Hello, thank you for having us here. It's a great honor for us to speak here and to participate in this conference. So we are now starting the first session of Quickfire pitches, and we have 3 speakers. The subject is life sciences and biomedicine. So I will be the first speaker. My name is Itai Katzir. I'm from Tel Aviv University, from the School of Biotechnology, Life Sciences. And I will show you a bit of my research that basically I'll show you how I am taking inspiration from biological systems and creating molecular function for biotechnological uses. Okay, so let's see, wait a minute. Okay. My title is Tuning the Dynamics of Viral Factories-Inspired Compartments Formed by Peptide RNA Liquid-Liquid Phase Separation. So basically, liquid-liquid phase separation, or LLPS in short, is a process where certain molecules interact in liquid solution to create these condensed phases that have unique properties. These can occur due to many types of non-covalent interactions. And basically, honestly, I think this subject is fascinating because you have so many challenges in studying these liquid structures. It's so hard to measure them. And there are so many unanswered questions in this topic. So LLPS, in short, has many examples in life and in eukaryotic cells in the form of membraneless organelles. Membraneless organelles are intracellular liquid condensates that have specific function and building blocks. There are many examples. For example, the nucleolus inside the nucleus, which is responsible for ribosome assembly. So what's common to all of them is that they are mainly made of intrinsically disordered proteins and RNA. Okay, so but we were intrigued by another type of condensates, and those are viral factories. And many types of viruses, including coronavirus, like in this image, measles virus, rabies, and many more, when infect their host cell, create these liquid condensates inside the cell that promote viral RNA synthesis, capsid assembly, many more processes. So we were intrigued, fascinated by these structures and how they are enhancing viral processes and act as actually microreactors. So we wanted to mimic that and create a minimalistic version of these viral factories that we could imply and add a function to create some kind of a biotechnology. So that's why we've made this measles viral factories-inspired system. So measles viral factories are mainly mediated by 2 proteins. One of them is the phosphoprotein that has this intrinsically disordered region in blue. And it is known to be crucial for the liquid assembly of measles viral factories. So we aimed to design a peptide that would minimalistically mimic the function of the P-loop in the liquid condensation of measles viral factories, and actually create a synthetic system that is inspired by these viral factories. So we've designed a peptide. We took the most prevalent amino acids in the P-loop sequence, and we have designed a peptide with spread glycines along the sequence. Those promote backbone flexibility, and it is known to promote LLPS. And we've made this peptide cationic with lysines and arginines, so it could interact electrostatically with the negative phosphate groups of RNA, just like in the viral factories. Okay, so this is the peptide viral factory-inspired peptide, VFP1 in short. It is 14 amino acids long and +4 charged. So it actually does interact with RNA and condensates into these type of liquid condensates. And we found that it can encapsulate materials and biomaterials quite efficiently. So we first wanted to understand what is the significance of each amino acid in this peptide sequence to LPS propensity. So we've made this alanine scanning test where we synthesize 14 new peptides. Each of them has one amino acid replaced with alanine, which has a simple methyl group side chain. And we've tested them all for LLPS with RNA, and basically all of them have generated LLPS except for 4 peptides, and those are the cationic replacement. It takes only one cationic amino acid to be replaced with alanine, and LLPS is completely arrested in any condition, any concentration. And this is because electrostatic interactions in the system are extremely crucial. You cannot infer the balance of them. So we have this system that encapsulates materials. That's OK. That's fine. But we wanted to add to control that encapsulation in a way so we could use it for biotechnological uses. So we've incorporated in the peptide this photoprotected lysine. So this is a lysine residue that is covalently bound to the NVOC protecting group that can be cleaved off with UV light. OK? So this is the VIF-P1 sequence. This is the lysine number 3, which was substituted with the NVOC protected lysine. And when this new peptide is subjected to UV light, the cleavage reaction occurs, and the lysine residue is exposed. So the peptide returns to its VIF-P1 form, okay, the original form. Remember that for later. So when we substituted this lysine with alanine, LLPS was arrested at any condition. So what happens now? With this peptide, the protected peptide, it actually does interact with RNA to create also condensates. But this time, they are much stickier and denser. And this test shows this perfectly. We test the LLPS propensity of both systems in increasing salt concentrations. And in black, you can see that the VIF-P1 system goes down eventually to 0, which means that LLPS is finished, no more LLPS, because salts mask electrostatic interactions. And the NVOC protected system in red is not very much affected. And we believe that this is because the aromatic moiety, the NVOC aromatic moiety, allows new modes of interaction to this system in the form of cation-pi interactions, pi-pi interactions between peptides and RNA. And this creates condensates that are much stickier and denser and are less diffusive because of these new forces in hand. So this fluorescence recovery after photobleaching test shows these changes in dynamics. We have 2 condensates, NVOC and RNA condensates. On the left, it was not subjected to UV. On the right, it was subjected to UV, and therefore the cleavage reaction occurred here. And when we bleach the center of the fluorescent RNA inside, we see that the diffusion on the right is much faster than on the left. Because of there is more of the VIF-P1 form, the electrostatic type form in this system. And we see that with increasing UV exposure times, we can gradually get faster diffusion, which is cool. We can tune the diffusion of the system with UV exposure. And these changes in dynamics also affect the encapsulation efficiency of the system. So we encapsulate, in this case, rhodamine B. And the NVOC system in red, without UV treatment, has a much higher encapsulation efficiency of rhodamine B. And after UV exposure, it drops down and gets much closer to the encapsulation efficiency of the VIF-P1 system. So now these changes in dynamics and encapsulation efficiency allows us to have these NVOC-protected compartments that encapsulate some material. When we irradiate them with UV, this change in dynamics causes them to release their cargo to the environment. This was quite crazy to find out at first. And you can see it right here. We measure the fluorescence of rhodamine B in both systems, the VIF-P1 system and the NVOC system. You can see in the moment of irradiation with the confocal microscope of a powerful UV laser, we see only a drop of fluorescence only in the NVOC system, which means there is a release of fluorescent cargo from the compartment, and this is not bleaching. So we believe this is a proof of concept for a potential light-triggered drug delivery system in the future, maybe. We have many more to study, and I'm trying to study it with living systems, with cell cultures. And if you want to hear more later about how RNA structure affects LLPS and how we can thermoregulate phase separation with these RNA and DNA structures, ask me about later or come talk to me. And that's it. Thank you very much.